Hey everyone, welcome back to another MCAT Shemmassian workshop. Today we're exploring a high-yield biology and biochemistry MCAT passage pertaining to receptor signaling, post-translational modification, and experimental interpretation. You can actually find over 4,000 MCAT practice questions on our MCAT question bank, but today we're just going to cover a couple of questions. We're going to read the passage paragraph by paragraph, discuss figure 1, and then we'll get to all 5 of the questions, complete with some explanations. Let's get started. So the way that we're going to be running through this passage is we're going to go through the paragraphs one by one, and I'm going to try and focus in on main ideas, key things that I would highlight, and if there's any particular concept or topic or content that's kind of ingrained in the passage, I'll bring those up and kind of give you suggestions on how you should further study those topics. So, starting with paragraph 1, it says granulocyte colony stimulating factor receptor, GCSFR, is a transmembrane cytokine receptor involved in the production of neutrophil granulocytes. Mutations in these receptors have been discovered in cancers, such as acute myeloid leukemia, AML, and chronic neutrophilic leukemia, CNL. The aberrant signaling mechanisms of these GCSFRs, however, are poorly understood. So this paragraph is pretty much just giving us some background on these receptors. There are these receptors, they're transmembrane, they're involved with the production of neutrophils, and having mutations in these receptors lead to really bad conditions, which should make a lot of sense, right? We have these receptors, they have to do with immune cells, like neutrophils. And if you have something wrong with it, some type of mutation, it leads to a condition. So things that I would highlight here would be receptor. It's a transmembrane cytokine receptor, and it's involved in the production of neutrophils. I would also highlight mutations. So again, the things that I've highlighted here are just things that kind of emphasize that this is a transmembrane cytokine receptor. It helps with immune cells like neutrophils, and there's these mutations that we're going to discuss, likely in the further paragraphs. On to the next paragraph. It states, scientists hypothesize that understanding the downstream protein targets of mutant GCSFRs might provide information about how GCSFRs contribute to cancer. The researchers developed an assay to detect levels of pSTAT5 and PERK1/2 in the presence or absence of GCSF for wild-type GCSFR and mutant GCSFR. The results are shown in figure 1. So the way that I read that, it could sound a little bit complicated, right? There's a lot of numbers, there's a lot of letters. Let's try and read this in a more simplified version. There's some scientists, and they believe that if they understand the downstream targets, protein targets of mutant receptor, that might provide information about how the receptor would contribute to cancer. The researchers developed an assay to detect levels of pSTAT5 and PERK in the presence or absence of stimulating factor for wild-type receptor and mutant receptor. So you see, the thing that I did here is, rather than reading all these numbers and letters, I just focused on the key thing that each one is. So what I did is, rather than say GCSF, I just call it a stimulating factor. Instead of saying GCSFR, I'm just saying it's a receptor. I'm keeping it to the bare bones of what each one of these things are. And that's going to make sure that I focus on the function of each one and that I can picture each one of these things. So if there's anything that I'm going to highlight here, it would probably be downstream protein targets of, and again, I would only highlight pretty much the letter R for receptor, GCSF-R. I would highlight the R. I'd highlight downstream protein targets, mutant R, mutant receptor, might provide information about how the R, the receptor, contributes to cancer. They detect levels of pStat5 and PERK, and then I would highlight in the presence or absence of SF, stimulating factor. So that's about all I would highlight. Again, this paragraph is simply just trying to tell me there are some downstream targets of this mutant receptor. If we understand those targets, we might understand how we lead to cancer, probably instead of neutrophils. And so they developed these assays, and they're looking at the presence or absence of a stimulating factor. A stimulating factor just turns a receptor on, right? Like a receptor is just looking for the stimulating factor. So whether that stimulating factor is present or not would have likely a very strong effect on that receptor. And that's probably going to be the basis of what figure 1 is going to be about. Okay, so jumping into figure 1, the figure title is that target phosphorylation of proteins of interest under different conditions. So the key things here are phosphorylation of proteins, and it's under different conditions. If you recall, in paragraph 2, it told us pretty much what those conditions are, right? It told us that we're looking at levels of pStat5 and PERK, and we're looking at it in the presence or absence of stimulating factor. We're looking at these downstream protein targets in the presence or absence of a stimulating factor in relation to, is it the wild type receptor or the mutant receptor? So looking at the graph, we can divide this into a couple different things. We can see the WT, that stands for wild type. That is your normal. That is your standard. That is your unmodified receptor. You also have T618I. T618I is just telling us that we have a mutation of the T amino acid at the 618th position, and it's being replaced with an I. So we have our wild type and our T618I. We also have our designation for pStat5 versus PERK. So we have pStat5 at the top and we have PERK at the bottom. And then our different conditions here are GCSF. You can see that there's 3 different rows for each protein, pStat5 and PERK. The first row shows a minus sign. A minus sign is just telling us that the stimulating factor is absent. Again, just exactly how paragraph 2 had indicated. And when we look across the board, we can see that without the stimulating factor, there seems to be little to no phosphorylation at all in those proteins. And you can tell phosphorylation levels based off of how dark the blotches are, how dark the blobs are. And so without the stimulating factor, we can see that there really isn't that much phosphorylation going on at all. The next row is this plus 12.5 minutes. That's telling us that the stimulating factor has been present. So the plus tells you that it's present. So the stimulating factor has been present for 12.5 minutes. And when we look across now for pStat5 and for PERK, we can see that the phosphorylation levels are now present. They are darker blots. They are darker bubbles, right? Like we have for wild type especially, it's very, very dark. So there's a lot of phosphorylation going on. For the mutant version, it's a little bit less, but there's still some phosphorylation going on. And lastly, we see the plus 90 minutes, which again tells us that the stimulating factor has been present for 90 minutes. And so we can see that as the time increased from 12.5 minutes to 90 minutes, with the stimulating factor being present, we can see that the phosphorylation levels started to decrease. So when you're looking at figures, you don't want to spend too much time looking at all these variables and all these different scenarios of with these conditions of things being present, not being present. You're looking for simple trends. That's what you want to search for. So in this case, if we look at all 4 of these scenarios, wild type, pStat5, mutant, pStat5, wild type, PERK, mutant, PERK, and all of these different conditions, we can see something pretty uniform. For the most part, when the stimulating factor is absent, there seems to be practically no phosphorylation. When the stimulating factor is present at 12.5 minutes, there starts to be phosphorylation for almost all of them, except mutant p-STAT5. At 12.5 minutes, there seems to be a lot of phosphorylation. Again, that was the only exception, but for the rest of them, there seems to be a lot of phosphorylation. And then as time increased, as it went from 12.5 to 90 minutes of the stimulating factor being present, it seems like the phosphorylation levels cut down in pretty much all cases. Again, maybe the only exception being mutant p-STAT5. Because the darkness levels, like the darkness of the blots between 12.5 and 90 minutes for p-STAT5 mutant seems to be roughly the same, if not maybe just a little bit lighter. So overall, as time increases with the stimulating factor being present, there is a decrease in phosphorylation. Without the stimulating factor at all, there seems to be practically no phosphorylation. And when the stimulating factor is present at the very beginning, there seems to be a fairly large amount of phosphorylation going on. So that's about as much time, maximum as much time you'd want to spend on a graph. Again, maybe a couple seconds. You're just simply trying to look at the title, look at the variables based off of the title, the key variables that are rooted in the title, and then you're looking for trends. And so if the trend doesn't stand out, very, very easy to you, then just move on. Just know what information, what material, what content, what variables are in the figures. That way, if you get some questions on it, at least you know what figure to refer to. In this case, the trends seemed pretty apparent. Again, large amount of phosphorylation at 12.5 minutes. As time increased to 90 minutes, phosphorylation decreased, and practically no phosphorylation when the stimulating factor was absent. And so now we're going to go into our final paragraph. Paragraph 3 says, in subsequent experiments, the researchers tested other GCSFR mutants, including a Q741N mutant, where N indicates any of the other 19 amino acids. They found that some substitutions led to increased signaling activity, while other substitutions decreased all signaling activity. In the future, the researchers hope to find new downstream targets of cancer-relevant GCSFR mutants. Again, if you wanted to simplify this, just read it without all the letters. Read it with just the key thing that each one of these things are. I would read that in this way. In subsequent experiments, the researchers tested other receptor mutants, including a mutant that has a change in the Q amino acid. That change in the Q amino acid could be any other amino acid. It could be polar, it could be non-polar, it could be basic, it could be acidic, it could be positively charged, it could be negatively charged. It could be any other amino acid. And they found that some of these substitutions led to increased signaling and some of them led to decreased signaling. This paragraph, I would say that there really isn't much to highlight here. It's giving us very basic, very broad information. We have a receptor mutant. There is a mutation happening. We don't know what the mutation is going to turn into, right? The N could be any other amino acid. And when that mutation happens, it can either lead to increased signaling or decreased signaling. So it's not giving us a ton of information. And so in reality, I really wouldn't highlight much at all over here. Again, all I would do is simply stamp this paragraph with the fact that there are other experiments going on, and there's this new mutation of Q741. I would simply highlight that just so that I know if there's a question or an answer option referring to that specific mutation or these subsequent experiments, I at least know exactly what paragraph that's referring to. So now that we've finished reading the passage and going through the figure together, we're going to go straight into these questions. But before we do that, let's just say that if you're looking for more information on MCAT buzzwords, do check out the comprehensive list of high-yield key terms listed at the bottom of each content unit in our MCAT Premium Content Hub. It's super valuable, and I think it'd give you great insights on just key words to be looking out for. That way, when you're reading these passages, you're just able to almost distinguish them. It's almost like a light in the darkness, right? It's just a flash of content that you should know, it should give you some background. It should give you some standing on what topic we're even talking about. And I think that'll help a lot. So with that being said, let's go into the questions. All right, let's jump into question number 1. The question states, one researcher hypothesizes that GCSFRs activate the MAP kinase pathway. Which of the following protein residues is most likely to be altered by this pathway? So you might be reading this question and asking yourself, what is a MAP kinase pathway? Where did we talk about that? So here's how I would read this question in a simpler way. I would say, one researcher hypothesizes that receptors activate the kinase pathway. Which of the following amino acids or residues is most likely to be altered by this kinase pathway? So this question, it kind of sounds like you would need to go back to the passage for this, but in reality, all it's asking is what amino acid would be affected by a kinase pathway. And hopefully remember that kinase pathways phosphorylate things. So it's just asking what amino acid would likely be phosphorylated. And in this case, there's 3 amino acids that can be phosphorylated. Any of the ones that include an OH group. In this case, it's going to be serine. And this would be a perfect example of a question that has a ton of content surrounding it. There's questions about, there is hints of information about receptors. There's hints of information on different types of enzymes and enzyme classes like kinases. And then there's these amino acids. So if you're doing this question, you wouldn't want to just say you got it right, but you just knew this serine could be phosphorylated. That doesn't mean that you don't need to know the other amino acids. That doesn't mean that you don't need to know the 3-letter abbreviation, the single-letter abbreviation. It doesn't mean that you don't need to know how to draw the structures of these amino acids. They're just different classifications. It doesn't mean that you don't need to know, if you knew kinases, that doesn't mean that you don't need to know all the different types of classes of enzymes and their examples, like transferase, phosphatase, phosphorylase, ligases, right? So there's so many examples of other things that are surrounding the content in this question. And that is really important to make sure that when you do these questions, you're doing this zoom out. The reason why you're doing that is because this question asked about kinase and phosphorylated amino acids. Next time, it might be something different. So if there's content and there's a lot of content surrounding that, make sure that you feel comfortable with all the content. Let these questions be identifiers for weak points in your knowledge. Question number 2 states that, what is the effect of the mutation on pSTAT5 activation based on the results from figure 1? So before I ever go into these questions with pretty long answer options, before I go back to the passage or back to the figure to try and find this effect of this mutation, I always like to look at the answer options and see if I can simplify things. See if I can narrow things down. I'm looking for similarities, I'm looking for pairs, and I'm looking for differences. I'm also looking for extreme answer options that might stand out and kind of make things just false. I'm also thinking about logic, right? What if there's a question asking about something being spontaneous? And there's 2 answer options with a Gibbs free energy that's positive and 2 answer options that's got a Gibbs free energy that are negative. If it's a spontaneous reaction, you could immediately cross off the 2 positive ones. And so there's a couple of things we're looking for answer options. We're looking for logic. We're looking for similarities, pairs, differences. We're also looking for extreme answer options as well. And so looking at the answer options, there should be 2 pairs here. Hopefully you've noticed. And the 2 pairs here are, 2 of them talk about less phosphorylation, and 2 of them talk about decreases or no decreases to binding affinity. So if we can figure out if figure 1 has to do with phosphorylation or with binding affinity, we can narrow this down to 2 answer options right off the bat. So going back to figure 1 now, we can see that target phosphorylation of proteins of interest under different conditions is the figure title. So this is telling you that it's about target phosphorylation. So then we could cross off C and D for that reason. Now we're simply down to A and B, and between A and B, there's a key difference. A says that there's less phosphorylation of the mutant at both time points when compared to wild type, and B states that there's less phosphorylation at only the early time point when compared to wild type. Looking at the figure, we can see that at both time points, the 12.5 and the 90-minute, there seems to be phosphorylation levels that are changing. So at 12.5 minutes, we have a really dark blot for the wild type receptor. For the mutant receptor at 12.5 minutes, it's a little bit faded, but it's there. So compared to wild type, the mutant receptor certainly is less phosphorylation. So we know the early time point is a checkoff, right? So we know that B for certain is that part of it is true. But what about both time points? What about the later time point? Is that part true? And so when we look at the 90-minute time point, does the mutant receptor look less phosphorylated than the wild type receptor? And the answer to that would be kind of hard to say, right? Those blots look fairly similar. The mutant receptor might look a shade or two darker, and maybe you could argue that there's more phosphorylation going on, but you probably definitely can't say that there is less phosphorylation going on there. A rule of thumb with figures, unless there's some type of significance being presented, whether it's error bars, whether it's p-values, whether there's stars going on, if there's none of those available, you have to go based off of your eye. You have to go based off of logic. And if there is no clear difference, then there isn't. So for the 90-minute mark, I would say that there is not less phosphorylation. There is not significantly less phosphorylation. So A would be false, B would be the correct answer. Question 3 says, what amino acid residue is most likely to be found in the transmembrane region of the mutant receptor? Again, this is one of those questions pretty similar to A, where it does reference things in the question that were found in the passage. A big point of confusion for a lot of students is whether that information is required to be pulled from the passage. So people are sometimes iffy on, am I supposed to go back? Am I not supposed to go back? Is this based off of content? And I would just be asking yourself, would you be able to simply answer this based off of the information that you know? And a lot of that usually comes down to simplifying and rewording the question. So the question says again, what amino acid residue, so which amino acid is likely to be found in the transmembrane region of this mutant receptor? That question alone gives us some very key information. And if you recall, if we were to even go back in paragraph 1, it did give us a little bit of information about that receptor. The only thing that it said is the same thing here. It is a transmembrane receptor. And so transmembrane being the key thing. Transmembrane should tell you that the amino acids that would likely be found over there would be nonpolar. And so if we're looking for nonpolar amino acids, you want to be thinking of these 4 options, which one of these would be nonpolar? And the answer in this case would be A, alanine. Let's move on to question number 4 now. Question number 4 states, figure 1 demonstrates that in the absence of the stimulating factor, the wild type receptor cannot do what? Again, this is a question. It uses a lot of keywords that did show up in the passage and that figure. And then another question is like, do we need to go back? right? There was a paragraph that did talk about the stimulating factor. Paragraph 2. It did talk about the wild type receptor. Paragraph 2. And then we also had that figure that, of course, had the stimulating factor being absent and the receptor and the different types of receptors. And the question is, do we need to go back? I would say no. If we were to reword this question, we should be able to simplify it in a way where we can answer this based off of content and logic. The question, again, is just saying, in the absence of a stimulating factor, a receptor cannot do what? So we're just simply getting asked, pretty much, what does a receptor do? If the stimulating factor is absent versus present, what could that receptor do? If we look at the answer options, we're doing the same thing as before, right? We're looking for answer options that have extremes. We're looking for answer options that have logic or don't have logic. We're looking for similarities, pairs, and differences. And if we did that, we should see a pair between A and B. A has to do with directly phosphorylating its targets, and B has to do with activating downstream signaling to phosphorylate its targets. So the pair here is that they both have to do with phosphorylating. The difference is that one directly phosphorylates, the other one activates downstream signaling to phosphorylate. So one's direct, one's indirect. C has to do with producing neutrophils, and D has to do with suppressing transcription. And so we're going to go back to the main root of the question. In the absence of a stimulating factor, a receptor cannot do what? A stimulating factor is just kind of like a ligand, right? It's just a signal. And a receptor, all that does is it just recognizes if a signal is present or not. Once it recognizes that the signal is there or not, it activates downstream signaling. It does not directly phosphorylate things. It does not produce different types of cells. It does not suppress transcription directly. So the key answer, or the key thing here, was whether it was direct or not. And the only answer option that alluded to some type of indirect process would be B. And so we're going to go with activate downstream signaling to phosphorylate its targets. On to our very last question. Question number 5 states that a researcher is attempting to understand the temporal trend observed in pStat5 or PERK 1/2 phosphorylation. RT-qPCR might show upregulation of which type of enzyme? Let's just kind of go over this question, but let's do the same thing as before. Let's simplify, let's reword, let's see if we can get to the root of the question. So the question's saying there's a person, and that person is trying to understand the time trend observed in protein phosphorylation. We're using reverse transcriptase qPCR, some type of lab technique. And if we used that lab technique, it might show an increase in upregulation of which type of enzyme. So again, let's just try and reword it even again. Let's try and even simplify it even more. If we're trying to look at the time trend of phosphorylation, a certain lab technique might show an upregulation of which enzyme. So let's even simplify and reword it again. If we're looking at a trend in phosphorylation, what enzyme would we see an increase in regulation? Or what type of increase in what enzyme would we see? The key thing in this question here is that you're looking at phosphorylation. And so there's two things that you could do here. You could either answer this question simply based off of content knowledge and a couple of tricks. You could also answer this based off of the figure. Let's do this the more correct way. I'll put that in air quotes, the correct way, which is by using the figure. And if you do recall, where we're going to say that, remember, we wrote this trend out, which was that if GCSF was absent, we practically saw no phosphorylation. When it was present in the beginning at the 12.5 minute mark, for 12.5 minutes, there tended to be a lot of phosphorylation in most cases. And then as time increased to the 90-minute mark with the stimulating factor being present, phosphorylation started to decrease. So as the temporal trend went on, phosphorylation decreased, therefore removal of phosphate. The only enzyme that has to do with removing phosphates is going to be answer option B, phosphatase. But I promised you another way of answering this. I promised you a way of answering this with content and logic. And so let's go over this. So a little trick to throw out is that for the most part, if a question on the MCAT involves phosphate groups, you always want to pick something like phosphorylase, phosphatase, or kinase. While transferases can move phosphate groups, and examples of transferases are some of those three enzymes. If it's involving phosphates, almost always you want to pick one of the three specific ones like phosphatase, kinase, or phosphorylase. The reason why is because it's more of a understanding of transferases, transfer functional groups, but these other three enzymes are specific to phosphates. So if we're going based off of simply that logic, if we're looking at trends with phosphorylation, the only enzyme here that involves phosphorylation is a phosphatase. A transferase, again, transfers functional groups. Oxidoreductase has to do with oxidation reduction, and ligase is an enzyme that puts things together. So the only enzyme here involving phosphorylation is phosphatase. So again, what we're looking at here is a set of questions where if we were to take a step back and if we were to look at this passage as a whole, we would say this passage it had a lot to do with phosphates, it had a lot to do with enzymes, amino acids, immune system, figure analysis of Western blots. So this passage involves a lot of content. And whether you're confident in the questions, maybe you might have done great on the questions, but maybe the other things that were surrounding these content topics you were feeling a little bit weak on. Like let's say that the question asked you about a different enzyme class instead of phosphatase or transferase. What if it asked you about a different enzyme besides a kinase? What if it asked you about different amino acids that instead of being non-polar, it was the polar ones? What if instead of, for question 1, it was asking you about amino acids that were basic instead of ones that could be phosphorylated? Would you be able to answer those? What if it asked about, instead of neutrophils, basophils, right? You just want to know, like when you finish these passages, they are just almost like little pins and they're showing you little bits of content. You take each one of these pins and then you zoom out on the concept. If you feel good on the concept, great, move on. If you don't feel great, read the explanation for each question, understand the content related to each question and all the answer options, and then ask yourself, did I make any strategic errors here? Could I have narrowed down any of these answer options to 2? Could I have knocked one of these options out? Could I have gone from a 1 out of 4 chance to getting a question right to a 1 out of 3? How about a 1 out of 2, right? If you're crossing off 2 answer options. So once you learn the content related to this passage, you zoom out and you cover the topics that you might feel weak on the concepts that are surrounding these concepts. Your zoomed out content review. And once you identify any key strategic changes that you could have done and that you can apply to future passages, that's when you're done reviewing this passage. And with that, it looks like we survived to the end of the passage. That was a pretty dense one. Thanks for joining, and we hope this walkthrough is helpful in your MCAT journey. If you're looking for more practice resources, you can visit our MCAT question bank or premium content hub for thousands of sample problems. Or if you want a more personalized approach, our MCAT experts provide one-on-one tutoring, as well as individualized study schedules and test-taking strategies that go a step further than just content review. Check out our website to learn more, or feel free to drop a comment here with any questions you may have. Happy studying.